So the next point, it is endospore. Now, basically we have to discuss that what is mean by spore and what is endospore and exospores. Now, first, bacterial spore. What that bacterial spore and why they are formed? Basically, the bacterial spore, they are highly resistant. Okay, in this description, the point it is indicated by specialized resisting cell forming by certain gram positive bacteria. So first point, it is resting. Specialized resting means those bacteria, they are having a resting position, means they are quite inert in a nature, but why those spores are formed. Now in the description, now what I am telling, it is not written in this diagram, sorry, it is in a slide, but listen it properly. The bacterial spore, those bacterial spore, they are highly resistant in nature, they are dormant in a structure, then they are no having metabolic activity, and they form in a response to the adverse environmental condition. Here in this description, what I am saying, you have to again read it properly or listen it properly. They are highly resistant. Those endospores, when they are formed only if there is a adverse condition in the environment observed, then and only then they will form. But when they will form, they are having a huge properties. They are dormant in a structure, they are more stable in a different types of environment, so they are termed as a dormant. Next, it is they are not having a metabolic activity as compared with your normal cell. They are easily metabolized, but the endospores or different types of spore, they are not easily metabolized. Then they do not have role in a reproduction. Okay, so as we know that if they are not metabolized, then they will not be part in a reproduction process. Then only the note, essential note, it is only the category, it is fungus, they are forming a role in a reproduction. Okay, if we are comparing the bacteria and fungi, it is a category. In this category, the cells are formed and the spores are formed in a fungus, they are participate in a reproduction. Okay, so these are the few properties of your spores. Okay, now the spores are specialized in a resting cell forming by certain type of gram positive bacteria. Okay, resting means they are more stable, they are more dormant in a nature. So example, it is genus Bacillus and genus Clostridium. These are the example. They are forming the different types of spore. Then they are highly durable dehydrated cell with thick wall and additional layer. Okay, here they are termed as, whenever the spores are formed, they are more durable, means they are more resistant to the different types of bacteria and environmental condition. Then they are having a thick cell wall and additional layer, can survive extreme temperature. Now what we discussed first, spores are formed because of the adverse environmental condition, and when spores are formed, they are termed as endospores, and they can survive in an extreme condition, means it is just a mutation of that type of some bacteria, and they are more or extremely survivable in any type of condition, so they termed as a survive in a extreme temperature. Then against your disinfectant, acid, base, they are having lack of water and toxic chemical and radiation. See, whenever the bacteria, they are having the lack of water, means they are more durable in nature. Now example, the endospore of some thermophilic bacteria can be survive 19 hours of boiling. Can you imagine that? 19-hour boiling, still those spores are surviving. So it is very huge a problem. We can observe that if the bacteria they are form the spore, they will largely survive in the environment, and it is very difficult to inhibit the growth of those spores. Okay, so the concern of food and health industries. See, imagine if this type of spores are observed in specifically food and health industries, so it is very dangerous for the human life. Okay, we can imagine. So these are the endospores. In this diagram, we can observe the endospore, the diagrammatic representation. Here the point B, we can observe that the spore they are having the core wall, then they are having the exosporium, then spore coat, plasma membrane, cortex, all those different types of parts are observed in that spore, endospore. But in the diagram A, you can observe the spore, endospore, it is present in a middle, then second one it is present in a top, the third one it is slightly in a middle or initial part of that cell. So on the basis of that, those spores are having spores and endospores, they are having a basic different types of shape, and on the basis of they are classified in the three different classes. Means if the spore it is spherical and it is central position, that is one of the class. Might be your spore it is oval shape and present in a central position, and they are non-budding. Okay, non-budding. In a second diagram, we can easily observe it is start to bud, means it will form a next position. So they are budding or non-budding. Okay, so if it is present in a at specific end, one end, they are termed as a budding. If it is center endosperm, it is spherical, then spherical and non-budding. So this type of simply classification observed in that type of endospores. Okay, now in this slide we can observe the process of your sporulation. Okay, now how those spores are formed. Okay, very important process. In this process we can easily identify that how those spores are formed. In that first step, it is new replicant DNA, it is isolated by the growth of plasma membrane called as a spore septum. Okay, don't worry about that discussion. First we have to see the diagram, and in this diagram we can easily understand all those 5 to 6 different types of step. Okay, so let concentrate on that point. We are discussing about the process of spore formation. We are discussing about the process of spore formation. What first process carry out? Okay, in this diagram we can easily observe this is a one bacterial cell, and first budding part. Okay, this is the budding part, means one additional part which is synthesized in the same bacterial cell. Okay, so means whenever budding it is started, at that time all the contents get divided into the two part, means cytoplasm divided, your DNA it is divided, your plasma membrane divided, your DNA content it is divided, all those components are divided. Okay, so first step of your spore formation, it is spore septum begin to isolate newly replicated DNA and small portion of your cytoplasm. Okay, so first step already we discussed, the DNA get replicated, it will form another budding area, and simultaneously small amount of your cytoplasm get divided into the two different types of part. Okay, now in the second step, what happened in the second step? The plasma membrane start to surrounded to the DNA, cytoplasm and membrane isolated in this step. In this step, step number 2, we can easily observe the budding part, it is isolated from the basic part of your cell. Now here it is totally properly surrounded by your cytoplasm and it is having the DNA. So this is second process, it is a simply formation of second one area. Now step number 3, it is spore septum surrounded isolated portion forming the forming the four spore. Now, in the discussion where we started, the word it is called as a four spore. It is four spore means another membrane, a second membrane, it is surrounded to that of one spore, that is your four spore. Okay, it is started to form in this area, so it is two membrane. Now it become a two membrane. Okay, so there are two membranes are formed in the step number 3. Now here in the step number 4, we can easily identify the peptidoglycan layer formed between this membrane. Now two layers are formed, and again that individual spore, it is again come into the basic cell. Okay, again come into the basic cell, and it will form another layer. It is a peptidoglycan layer. It is a cell wall layer, external layer it is formed, and this is the fourth step carry out. Third step we can observe second layer it is observed, and next one it is peptidoglycan, it will form the cell wall, and fourth step it is called as a spore coat formation. Step number 5, it is spore coat formation. Now already we discussed that in the before that slide, spore they are having multiple layers. Okay, so in the step number 2, step number 3, step number 4, we observed that the multiple layers are formed, and the last external layer it is spore coat. At spore coat it is formed, and after forming that spore coat, that spore get ejaculated from the host bacterial cell, where the bacterial basic cell it get killed, and endospore it is free from the cell. That is a last step carried out by your spore formation. So these 6 steps are observed in this diagram, and here we are discuss about the formation of spore. And in the next slide we can observe the depth process. Okay, again this 6 step process it is divided into multiple points. Okay, let me see the detailed step. Okay, so what are those detailed steps? Okay, so first one it is vegetative cell. Vegetative cell means your basic cell from where the spores get formed. Okay, in that wall, then cytoplasmic membrane and DNA, all those basic contents are there. Now the DNA more dense, first step. Okay, so now we are concentrating on the more specific step in that 6 different step formation of your spore. Now how those endospore get deeply divided? First step it is divided, start to divided from the vegetative cell. Now what first happen, the DNA it is become more dense. Okay, when your DNA become more dense, then stage first will form the DNA. Here we can observe in this diagram it is become more denser one. Okay, in that stage 2, what is the stage 2? It is cytoplasmic membrane, it is invaginated to form the spore septum. Okay, so in this portion we can observe the second one portion it is formed, and it is get separated in this position, that is cytoplasmic membrane get divided into the step 1. So detailed steps it is in that first it is separated from vegetative cell, it DNA become more denser, and then it get separated from your cytoplasm, that is step number 1. Now step number 2, what is step number 2 or for step number 3 portion? It is what? The spore septum grow around the protoplast, that is engulfment. Okay, now in a last diagram the 6 different steps we are observed. The what those the 6 step in that stage number 3 we observed that the again that spore it engulf into the host cell. Now here the process it is stage number 3, it is engulfment, means that spore again enter into the host cell. Okay, after that it will form a 4 spore formation. It will form 4 spore formation, means again additional one layer it is surrounded to that of spore. Okay, so here in the diagram we can observe the pink color portion, it is of your additional external layer of your spore. Okay, now in the stage number 5 we can observe that the pink layer it become more denser and it become dehydrated. It become dehydrated means it is not having a watery amount, so it become a exosporium. It become a exosporium. So why it is observed? It is because of more acceptability or more what we say that it is more dormant in the nature. Those things are observed in a stage step number 5. Okay, in the next step what happened, here we can observe stage number 5. It is having a coat layer to form. Here we observe the coat layer, and in the stage number 6 it what will happen, the maturation development of your resistance to the heat and chemicals. Here we can observe that the pink layer, okay, and surrounded by your black layer. So what will happen, it become matured. Okay, whenever that spore become mature, and how it become mature by your resistance regarding to your heat and chemical, and it will become a mature, and after lysis of your cell it get released and free from your spore. Okay, so step number 7. Now in the last the 6 step diagram we can observe the only superficial discussion, but in this all those 7 steps we can easily all observe the basic and minute step involved in this formation of spore. So stage number 7 we can observe that lysis of your cell and release form of your free form of your spore. So here in this diagram it is a free spore having a core as well as it is exosporium. Okay, so this is a free spore which can be easily surrounded and having the different types of property. They are more having dormant in nature, they are highly resistant in nature. Okay, so this is the simple diagram, final circulation of your specification of your spore. Okay, and the sporulation, the whole process of sporulation, they are having a properties. Let's see those properties. May be a part of your normal life cycle or triggered by the adverse environmental condition. Okay, here why that sporulation observed, but obvious it is because of adverse environmental condition. Okay, then the endospores do not carry out the metabolic reaction unlike normal vegetative cell. Okay, this is the property of your endospore. They are not carry out all the metabolic reaction as consideration of your vegetative cell. Means normal cell they are carry out all the reproductive and metabolic processes, but endospore though they are never carry out that vegetative process. Then endospore can remain dormant for thousands of years. Okay, so already we discussed that the dormancy period of your spore it get increases, and germination of endospores return to its vegetative state. Usually it occur when the environmental condition become more favorable and triggered by the physical or chemical damage to the spore. Okay, now here we can observe that those vegetative cells they are forming the endospore by sporulation process, and again when those specific spore endospore, if they get the proper favorable condition, here we can observe the center it become more favorable. Whenever the environmental condition it become more favorable, then and only then they will again become a vegetative cell. By the process it is called as a germination process. Okay, means first one it is vegetative cell. With the help of that vegetative cell it will form a endospore, and after sporulation process it will form a germination, and again it become a vegetative cell. Okay, so first activity vegetative cell it is a metabolically active. Endospores are not metabolically active, and again your vegetative cell are metabolically active. Okay, and this diagram we can easily observe the different endospores. Okay, here we can observe the red color and sky blue color. Okay, So the sky blue color they are endospores, and red color they are safranin staining your cylindrical bacilli. Okay, so here we completed the first part of your bacteria. Thank you.